Welcome to the third and final part of online Lecture 4. This part of Lecture 4 focuses on a unique and important metabolic pathway, photosynthesis. The term photosynthesis means to make from light, and during this process, light energy from the sun is converted to chemical energy in the form of glucose. This pathway is an anabolic process, whereas cellular respiration and fermentation, which were considered in part 2 of online Lecture 4, were both catabolic processes. This harvesting of the sun's energy during photosynthesis provides most of the energy that fuels life on Earth. However, although the process is anabolic and begins with energy from the sun, it has much in common with cellular respiration, particularly with regards to redox reactions and chemiosmosis, and also the breaking down of an involved metabolic process into several steps. Photosynthesis involves two pathways, the light reactions, and this is the step where light energy is converted into chemical energy, and then a second set of reactions called the carbon fixation reactions, where the products of the light reactions are used along with carbon dioxide to produce carbohydrates. This slide shows an overview of the photosynthetic process. In a manner that's analogous to what occurred in the mitochondrion, there are some processes in photosynthesis that occur within a structure called a thylakoid membrane, which has a double membrane type structure, and some processes that occur out into the open cavity of the chloroplast, which is called the stroma. So you can really think of this stroma as being analogous to the matrix of the mitochondrion, whereas the thylakoid with the double stacks of membranes being analogous to the inner and outer membranes of the mitochondrion. In terms of the reactions of photosynthesis, the light reactions take place in these thylakoid membranes, whereas the carbon fixation reactions take place in the stroma. During the light reactions, energy from the sun is absorbed and makes ATP and NADPH, and these are then used to drive the endergonic formation of sugars during a process called the Calvin cycle. Again, here's an example of that reaction coupling that we've seen throughout this unit on metabolism. Understanding how light energy is converted to chemical energy is one key to understanding photosynthesis. And in order to understand that, you need to understand some things about light. Light is a form of electromagnetic radiation, and it's very interesting because it travels as a wave but also behaves as a particle. The particle behavior is attributed to a packet called a photon, which is basically just a packet of energy. And these photons can be absorbed by molecules and add energy to the molecule. When this happens, it moves a molecule from what's called a ground state to an excited state. And what we're going to see during photosynthesis is that this movement actually involves the movement of an electron to a position a little further out from the nucleus. We've already seen that in the electron transport system that moving electrons have the ability to do work and carry energy. As I said before, light is a form of electromagnetic radiation, and for photosynthesis, we're actually only interested in a fairly narrow range of electromagnetic radiation. That is the visible light, which is between 400 and 700 nanometers. In order to absorb photons, organisms must have receptors to receive that signal, and these occur in the form of pigments. Pigments are molecules that absorb only particular wavelengths in the visible spectrum. To figure out what kind of wavelengths a pigment absorbs, the absorption spectrum for that pigment must be measured. This is measured by exposing the pigment to a variety of wavelengths of light and determining which one it absorbs. This is very different from something called the action spectrum, which actually plots how much biological activity there is at each of the particular wavelengths. Let's take a look at an example. This graph is showing an absorption and action spectrum looking at a plant called Anacharis. And if you look at the green line, you'll see the absorption spectrum of chlorophyll A, and you can see that it has two peaks, one around about 440 nanometers and another at around 660 nanometers. But if you look at the blue line shows you all of the wavelengths at which photosynthesis occurs by Anacharis. So what you could infer from this then is if there is biological activity at wavelengths that chlorophyll A does not seem to absorb light, that there must be other pigments that actually help absorb the light, and that is in fact exactly the case. These pigments are what are referred to as accessory pigments, and they basically absorb some of the wavelengths between the red and the blue and then end up transferring some of that energy to the chlorophylls. So the primary pigments that absorb light in plants are chlorophyll A and chlorophyll B, but they are helped out by some accessory pigments, as I just mentioned. The pigments themselves are actually buried down in those stacks of flattened sacs called the thylakoid membranes. The pigments are actually lipids, so they exist well inside the hydrophobic environment of the cell membrane. This figure shows the molecular structure of chlorophyll. And if you look down at the bottom of the screen, you can see a tail that looks very lipid-like and would obviously make this molecule quite at home in the hydrophobic environment of the membrane of the thylakoids. As I mentioned before, the role these pigments play in photosynthesis is that they actually absorb the light. And what happens exactly is that when this chlorophyll absorbs the light, it enters into an excited state, and then it rapidly returns to a ground state. But when it does that, it releases the energy in the form of an excited electron. And it's at this moment where that light energy has been transferred to a chemical energy in the form of an electron. And in a very similar fashion to what we saw in the electron transport chain of cellular respiration, this excited electron that has been taken from the chlorophyll molecule will actually transfer to an acceptor. The chlorophyll then becomes oxidized and the acceptor molecule is reduced. This initial electron acceptor is the first in an electron transport system. The final electron acceptor in this process is NADP, which is very similar to NAD except that it has a phosphate group. This NADP is an electron carrier analogous to NADH, so it is reduced, and its reduced form is shown here on the middle of this slide as NADPH. And when this reduction reaction occurs, what has happened is that the light energy that was absorbed by the pigment and then transferred to an electron is now bound up in NADPH, which can release that energy for cellular processes when it is oxidized somewhere else down the road. This slide gives you an overview of what happens during photosynthesis. First, we see the photon being absorbed by photosystem 2. It excites an electron which travels around until it reaches the final electron acceptor. At this point, the electron is donated to an electron transport chain, and that electron transport chain results in the formation of ATP. I'm not going to go over that in detail because it is exactly analogous to the process we saw in the mitochondrion. There are only a couple of things that I want to point out here. First of all, this chlorophyll molecule that donated its electron would stay in a permanently oxidized state unless it gained an electron from somewhere. And in fact it does, and it gains an electron from the splitting of water, which yields 2 protons, which are used to pump across the membrane to generate the proton gradient for ATP. But it also yields gaseous oxygen. And it is actually this step of photosynthesis that really changed the chemical environment on Earth from an anaerobic to an aerobic environment. So you have photosynthetic organisms to thank for the oxygen that you breathe, because without it, animal life on Earth would not have been possible. As I mentioned before, the ATP that is generated during the light reactions of photosynthesis is necessary to drive the steps that create sugar. These steps are referred to as the Calvin cycle, and it occurs in the stroma of the chloroplast. In an analogous similar to what we saw in the citric acid cycle, the Calvin cycle consists of a series of biochemical reactions, each of which is catalyzed by a specific enzyme. This figure shows the Calvin cycle, and the Calvin cycle can be divided into 3 primary phases: a carbon fixation stage, a reduction and sugar production stage where NADPH is oxidized and used to reduce the carbon-containing compounds, a sugar production stage, and then finally a stage that regenerates the compound that's needed for the carbon fixation step. It's shown here as RUBP, that stands for ribulose bisphosphate. We're going to look briefly at these steps in more detail. The first steps involve the fixation of carbon dioxide. What happens in this step is that gaseous CO2 is added to ribulose bisphosphate. And there is an enzyme that catalyzes this reaction that's called ribulose bisphosphate carboxylase oxygenase, or Rubisco. This fixation of carbon dioxide forms a 6-carbon molecule, which is quickly broken into 2 3-carbon molecules, which are called 3-phosphoglycerate. This slide is showing the process of carbon fixation and the generation of those 2 3-carbon compounds that we just discussed. The next step of the carbon cycle is the reduction and sugar production step. And in this case, the reduction is actually occurring to the carbon-containing compounds, where that 3-phosphoglycerate is reduced to form glyceraldehyde 3-phosphate. If you looked carefully, you saw that the energy to drive this step actually came from NADPH, which in this case is actually being oxidized. And remember that that NADPH was generated during the light reactions. In the final phase of the Calvin cycle, the electron acceptor ribulose bisphosphate is regenerated from that glycerol 3-phosphate. However, some of the extra glyceraldehyde 3-phosphate is actually exported to the cytosol and is converted to hexoses, glucose and fructose, for example. And eventually, when a plant accumulates enough glucose, it's actually linked to form starch, a storage carbohydrate. And as I mentioned before, this process of creating sugars from the energy that's initially in sunlight is responsible for most of life on Earth. So ultimately, for very few cases, the energy in almost all ecosystems can be traced back to some photosynthetic process at its base. This concludes part 3 of online lecture 4. The main points that you should be taking away from this lecture are that photosynthesis is a process of converting energy from visible light into chemical energy. You should understand how that first occurs through the excitation of an electron, and you should also understand how that electron traveling through an electron transport chain results in the generation of ATP, and also NADPH. Both the production of ATP and NADPH are analogous to the chemiosmotic process that we saw during cellular respiration. Finally, you should understand the relationship between the light reactions of photosynthesis and the Calvin cycle. And you should also have some idea of the overall steps that occur during the Calvin cycle. Again, I can't stress enough that this is a chapter that's full of many details, but if you start to look for the similarities and understand what it is that organisms need to do in order to fuel cellular processes, it'll give you a better conceptual framework in which to plug those specific details. This concludes the material that will be covered on Exam 1, which will be given in class on Wednesday, September 25th.